So, okay, let's start. So, again, thank you very much for tuning in. And I know you're all very busy and, it's always a bit of a challenge, but, that's really great that you're tuning in today. And today's speaker is Professor Michael Rychlik from TU Munich. And he's one of our international collaborators and also a honorary professor at the University of Queensland. And I have a bit more when about his bio in a few seconds. I think most of you already met me or know me. I'm Michael Netzel, a senior research fellow at the Center for Nutrition and Food Sciences at QAFI. And before we start the seminar, I want to do the acknowledgement of country. The University of Queensland acknowledges the traditional owners and the custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. Yeah, as always, first a few housekeeping things. Very, very simple. So the seminar is scheduled as always from 12 to 1 p.m. I guess Michael will finish maybe 12:50. So we have time for questions and hopefully answers. Please use the Q&A button, not the chat function, because at the end, then I will go through the questions in the Q&A function, and Michael will try, and hopefully I'm sure he can answer all your questions. Yeah, all your questions, whatever you think it's interesting or related to his presentation, just put it in and we will try to address them all at the end of the seminar. If you can't, not enough time, Michael is more than happy, then you can send him an email and will try to answer your question maybe by email. So yeah, what I mentioned is, today's speaker is Professor Michael Rychlik from TU Munich. TU Munich is one of the universities of excellence in Germany. And Michael is, not at the moment, I think since a couple of years, heading the chair of analytical food chemistry at TUM in Germany. He graduated in food chemistry at the University of Kaiserslautern, that's also in Germany, in 1988. And his PhD studies, it's very interesting, maybe a surprise for some of us, was on the flavor of bread and completed in 1996. And he was appointed as a full professor at TUM in 2010. In 2015, he served as a visiting professor at UQ here in Brisbane. And in 2016, he was appointed as an honorary professor at our university. He's also active as a visiting professor at the National University of Singapore and also teaching at the University of Hong Kong and the University of Chemistry and Technology in Prague, the Czech Republic. So they're really all very prestigious universities. And I think one more, I think that's really important. And since 2014, Michael is the head of the Committee on Contaminants in the Food Chain at the Federal Institute for Risk Assessment in Berlin, Germany. So that's a very important institute and monitoring, the food chain and contaminants and toxins. So that's a really very important role for Michael. I think that's all. I mean, there's much more to tell about Michael and his academic career, but I think we keep it, just keeping the time in mind. Would say, Michael, now it's all not the stage, but the Zoom is all yours. And yeah, please go ahead. And I'm disappearing now.
No, let's have a movie here. Yeah. So thanks a lot, Michael, for the kind introduction. And I'm happy to be here at the QAFI webinar, not seminar, but a webinar. And yeah, I'm happy to share our results on research with you. And I'm not going to talk about toxins today and a risk assessment, but about vitamins. And my talk will be about some water-soluble vitamins, B6, B9, and B12. So let's go ahead. So about vitamins and the research. So we have quite a lot of challenges here to tackle with. What we are working on in analytical food chemistry is about vitamin contents in foods, of course, but that's not the whole story. We have as well to consider vitamin bioavailability, and that is highly dependent on the vitamin 4. I'm coming later to that, because it's not just one for each group, not just one vitamin, or one structure. There are different ones, and that can be highly variable. And it's as well, the bioavailability is as well dependent on the food matrix and the individual physiology of the individuals that take up these vitamins. Right, my outline of my talk will be, so I'm going to start off with folates, analysis and bioavailability of this group of water-soluble vitamins. And then I'll continue with B6, B12, and have concluding it with an outlook. Right, so about folates, and if you have had the chance to listen to one of my talks before, so that's repetition, but if you haven't, so here giving you an introduction to folates in general. They are involved in methylations in amino acids, so amino acid synthesis and DNA synthesis as well, and it's a critical vitamin. It's a critical vitamin because the intake in Europe, at least in those countries where there is no mandatory fortification available, it covers just only 2/3 of the recommended daily intake. And if there is a deficiency or under intake, let's say that way, there are possible consequences of this deficiency, the most important ones, the neural tube defects. And as well, this deficiency is associated with an elevated plasma level of homocysteine, which has as well an implication on coronary heart disease, and as well some other aspects like colon cancer or Alzheimer's disease are maybe involved in the deficiency with folates. Problem of analysis of the folates is the variety, structural variety of this group. So we've got here on the slide, you see the structure of folates in general, and there are different variations here. What you can see on the here on the pyridine ring, for example, there could be different types of methyl or formyl groups attached to it. The oxidation state of the pyridine ring could be as well different, and the number of glutamides attached to the structure can be as well highly variable. So it means in reality, we got about 50 different compounds which all are vitamin active, so all have to be considered and all have to be analyzed here, and as well, and they are, well, as usually vitamins, they are susceptible to temperature, to elevated temperature, to oxygen, so on, to light as well. So it's really a challenging group. Right, what we are doing and what we found out is the best solution for these challenges is the stable isotope dilution assay, which is given here as a short outline of it, where the point is that you add an isotopologic standard here at the beginning of the analysis, which mixes with the analyte, and then during the whole process of analysis, this ratio, there is a certain ratio between the standard and the analyte, this won't change. Even if you lose some amounts of the let's say of the compounds, and so the ratio is unchanged. And you can differentiate between both by mass spectrometry, which means in the end you can, if you know the amount of the standard that you added at the beginning of the analysis, then you can just by simple rule of 3 then calculate back with the mass spectrometric differentiation to the amount of the analyte that is present in the sample. Right, example for the folates is just example here, the 10-formyl folate, which is one of these compounds that we are analyzing here. And we got here the unlabeled one and on the top, and the isotope log for analyzing it and for quantifying it would be the lower structure 13C labeled, 5-fold 13C labeled, which gives a different mass and makes it accessible to differentiation by mass spectrometry. And the point is that there are isotopolopes. That means they behave essentially really identically, chemically and physically. So there is, during the whole process of analysis, there is no separation or differentiation between both. Just at the end of the mass spectrometry, you differentiate them, and then you can calculate the ratio and the amounts. Right, here a little bit more, a better picture that you can imagine how that really works in reality. I mean, at the beginning, you put to the sample the standards, then you have some enzyme, let's say, enzyme treatments for matrix digestion and as well for the deconjugation of the polyglutamate chain. You need to, let's say, you need to really split off the chain until to the monoglutamate form, because we are only able to analyze the monoglutamate form. So for this we have the label standards available. There are still some purification of the extract with SPE, solid phase extraction, and then at the end you've got the LCMS measurement. Right. Chromatogram, LCMS chromatogram looks like this. So you've got here the five monoglutamides here. You see with each peak, you see two, let's say you have two signals and so on. The one signal for the analyte, the unlabeled one, and underlaid is the one for the labeled standard. You see they are really exactly on the same retention time, and so that it's rather easy to, let's say, to assign them to the standard and the analyte, and then it's rather easy by the area just to calculate the different ratios and then the amount. You see here the 5 ones that we are analyzing, starting with tetrahydrofolate, then following 5-methyl tetrahydrofolate, then 10-formal folate, 5-formal tetrahydrofolate, and folic acid. Yeah, what we added just recently to this method here is the 10-formal folic acid, where we got from our collaborators from Merck, we got the label standard, which hasn't been available before. Right. Coming here to some results and some amounts in food that we have been analyzing, and we are focusing a little bit on those regions and those foods where there is a huge global biodiversity, because we think that there are still unknown, undiscovered sources for folates, which may really be helpful, let's say, to fight the folate deficiency globally. And you see here, and we're looking at the regions of high global diversity is in the tropical, particularly in tropical regions. So we got South America, Africa, Southeast Asia, and of course in Australia we got particularly Queensland and northern Queensland with its tropical area. And here's a display of fruits that are available in tropical Queensland. Here's, you see lots of high diversity. Of course, not all of them. Their origin is in Queensland, but they grow all here. And as well, we have been looking at some, let's say, really native fruits and foods in Queensland. I'll come later to this. Right, just a table on, not to talk about much about those different foods, but just give you an overview which tropical ones we have analyzed and what we found out. So you are here on this table, you see the folate content, and then vice versa, you see the daily amount that would be necessary to consume of this fruit to reach the recommended daily intake. You see on the top there is the wheat bread, which you would need around 3 kilos of that, so unfortified wheat bread. So that's not really manageable here, really, to cover your intake with bread alone. It gets better with mango and with okra and passion fruit. So you need for passion fruit, for example, around 300 grams which is manageable if the passion fruit is available to you, but which might be really more attractive to you or accessible to you is strawberry with 260 grams, so which is a bucket of fruits. So that's really manageable, and I think any one of us loves and likes strawberries, so that is really it has a high potential. Avocado as well, and then it's getting more to the, let's say, the more those sources that are higher in folates, spinach, of course, and would like to highlight on the one hand durian, which is probably, you know, that's sort of the stinky fruit coming from Southeast Asia, which is really high in folate. So you would need only 120 grams of it. And I added here one rather recent source is the tempeh. It's a fermented soybean, which is as well high in folate. So you would need just 100 grams. So it's like tofu, for example, or bean curd, and it has a rather similar taste, and you can just give it as seasoned and whatever in type of food or dishes. And so we see quite a big potential in that. And at the bottom you see beer, and you see you would need 4 liters of beer to cover your folate intake. So that's not really manageable or recommendable, let's say it that way. Right, so that's about just some samples that we have been analyzing sometimes ago, but now I would like to go over to come to the Australian bush food here, and we see here the green plum has been analyzed by a PhD student that has been co-supervised by myself and by Michael Netzel and Yasmina Sultamava. Is here the green plum with total folate content of around 100 to 150 microgram per 100 gram fresh weight, which is in the range, in the upper range until to the strawberry. So means is as well a good, a valuable source for folates. The other foods or other bush foods here are the green ants, and we are lucky to see a picture of our last expedition to the rainforest, and we have as well been, well, not sampling, but we have been tasting some of these green ants. They are all over there, and you just may eat. I won't go into detail how you have to prepare or treat them, that you can eat them, but you can just, if you look at the abdomen, the green abdomen, which is really like rather acidic here and has a folate content of about, let's say in fresh weight, let's say about 100. So not too bad, but if you need to eat about, let's say 200 grams of these green ants, so that's not really manageable. So it's like a snack and not really a reasonable source. Right, we have been looking as well to other insects, because this is there's quite a hype in Europe about insects as novel foods, and we're looking at it about the potential sources for folates. And we have been analyzing here the house crickets, mealworm, field cricket, locust, and lesser mealworm, and they are all in the range in the fresh weight. What you see here, about 60 to 140. Well, not too bad, but where they are really light, those samples. So it's just like a snack, and you won't eat 200 grams of these. So it's, well, additionally value, but it won't solve the problem, not at all. Right, now about bioavailability of the folates, and which usually is done in, or can be done in human short time studies, where you give food here and then you analyze the plasma folate. And then with the plasma curve, you can then calculate the bioavailability with respect, considering a reference folate, which is often done with folic acid here. We give a reference dose, and then you, with the area under the curve, then you can calculate the relative bioavailability, where for folic acid is assumed to be 100%. But this well, we have been doing this research with Michael Netzel a long time ago, and as well some further studies. For example, for the strawberry here, which we did here on this, seen on this slide. So see here the folate-free control day. You see the supplement test day, which is now not folic acid, but 5-methyl tetrahydrofolic acid, where you see a nice plasma curve, which is the downside of this method, does not start with 0, because you don't have a 0 level in your plasma of folate. So you always start off with a level at time 0, which is not 0. So that's why the, let's say, plasma curve calculation, the area under the curve calculation, is rather difficult. For the strawberry test day, you see that as well a nice curve, and we calculated this with that approach, and we found out that the bioavailability or bioappearance, how you want to call it, is about close to 100% of the strawberry. So that's really good news here for those who like strawberries. So remember, a bucket of strawberries a day would cover your daily requirements for folate. Right, but I've been telling you so that the AUC, the area under the curve approach with the unlabeled folates here, is rather tricky here for the calculation. That's why we went further and we developed a triple isotope study, where I won't go too much into detail in this busy slide here, but I would like to highlight these two aspects here. So it's what we change now. So just the problem is to get really at the time point 0 a 0 signal. And this is only possible with isotopologues, because they are not naturally present in the body. And that's why we have been dosing those isotopologues. Remember, stable isotopologues, so there's no risk with it. And on the one hand, the food ones in the food, and how to label those. So you need to intrinsically label those isotopes in the food, and which we did with yeast here with the 15N labeling. And the reference dose is that compound which is assumed to be 100% bioavailable. We used an isotopologue which was newly synthesized by Merck, which we dosed as an isotope, which as well is not naturally present in the body, and that's why we thought, and it worked out, that is you just 0 signal at the time point 0. So first about the yeast labeling as the food model, and then how do you label yeast with isotopes? So the straightforward way is here to doing with 15N, because the yeast is dependent on 15N with ammonium in the media, and that's why we fed the yeast with 15N labeled ammonium chloride, and the yeast incorporated that in the whole, let's say, organic mass here, particularly as well the folates. So the folates were all, and we saw that in the mass spectrometer that the folates were as well fully labeled with 15N. So this was the one hand where the tracer for the food dose. Right. Question is, okay, you've got different isotopologues. So you've got here on the top, you see the 5-methyl, the unlabeled one. Then you got the 13C labeled one labeled in the glutamate chain. Here the second one, which is our internal standard for quantification, just for quantification, not for tracing. Then we got the 2 further ones. So the tracer isotopologue for the reference substance here is, and this one is 13C labeled as well, but differently labeled. It's labeled in the phenyl ring in the middle of the structure. And then we've got the labeled one in the yeast as the food model here, and that's 15N labeled. So you see here at the bottom. And so the question is, can you differentiate all of them? Yes, it's possible, although their mass difference could be rather low, is down to one Dalton, for example, but you can differentiate all of them because they have their label incorporated in different parts of the structure. And with the tandem mass spectrometry, where you analyze the different parts of the, or different fractions of the structure, you can differentiate them all rather easily, and which worked out, you see with this, let's say, yeah, peak on them, with all of them really co-eluting but differentiated really by the mass spectrometer. Right. And it worked well. We did this study with a short and a small pilot study with 5 volunteers. And you see, that's how we expected. The blue line here, the blue plasma line, is the unlabeled one. You see, so that's really hard. It would be really hard to calculate this. But the other 2 lines are, on the one hand, it's the food one, the yeast label here, and on the other hand, it's the reference dose. And you see really clear plasma curves where you can calculate the area under the curve, and then calculating the ratio, and then calculating the bioavailability. And we found out here that it was for the yeast, it was about, well, depending on the volunteer, of course, got around relative bioavailability of about 63% plus minus 10. So this is the individual variability of the bioavailability of the folates. So 60% is not too bad. The general assumption of the food folates is about 50%. Yeah, so we could confirm that we are with the yeast here in that range. Right. Leaving the folates, coming to the pyridoxin group, B6. Well, B6 is as well important in amino acid metabolism involved. We have different vitamers. So at the top layer, top you see the, let's say, the traditional ones, which you might know, with the pyridoxin and the respective phosphates. But what we have been looking as well about is on the glucosides here, that are generally, they are not analyzed. They are included in the analysis. They are split off by glucosidases, but we want to look into their direct quantitation. That's why we developed the respective internal standards for this and analyzed all these compounds, where you see here, which is an example for a wheat beer. As you may know, that Weinstefan, where our campus is located, is really close to the oldest brewery of the world. So that's why we are looking always on beer. And you see here the wheat beer, and you see the first peaks are the traditional vitamins here, and then it starts off with the glucoside. The PNG is the glucoside, which we expected as well, but there were as well some unexpected one. For example, the PN maltoside, which is here highlighted as well, and the PN cellobioside, which have really significant peaks and significant amounts, and as well some unknown glucosides as well, which we are currently looking at and trying to identify in their structure. All in all, if we do the same calculation here for B6 with some food that we have been analyzing, so you see as well with white rice, you won't cover it. So you would your daily recommended daily intake would just need 6 kilos of them. So that's not really manageable. Strawberry 1.4 kilo. Well, I'm looking to Michael. That's not well. A little bit too much, huh? We've gone to which was surprising to us is that broccoli is as well as a quite good source, which we would need 500 grams per day input. But what we found manageable probably is the potato with 400 grams per day. And of course, beer, we have been looking at beer, but you would need here 1.7 liters of beer, so which is as well hard to, well, for alcohol-free, even alcohol-free, which could be recommended, it's even hard. Right, and in this in this respect, I would like to tell you about one project that we are currently have been starting with respect to the TEMPE. Going back one here, the TEMPE is as well a rather good source for B6, and that's why we have been or still are looking in Singapore at our branch at TUM Create, where our institute or our faculty has as well installed a lab here and other food processing facilities where we are as well producing indoor soybeans and indoor algae here for, let's say, novel agricultural practices here. And as well, in this respect, we want as well to produce from these indoor soybeans as well the TEMPE as a really good source for folates as well and for B6. And if you are interested in seeing more about this project, just have a look at the website shown below. Right, and last vitamin group in my talk would be the B12. So B12, you may be aware, is as well a challenging one, because first the structure is complex, and then the amounts are really low. So we are not talking about milligrams per 100 grams. And so we are in the lower microgram per kilogram range here. But the structure, and as well, different vitamins. And we have been, what generally is done in vitamin B12 analysis is cyanocobalamine. It's just some method of it. And where it is assumed that all this, that it reflects, let's say, the total amount of B12 and bioavailability of this vitamin. But we have been as well looking or wanted to look at the different vitamins and which vitamins are possible. So I would like, if you look at the complex structure, you see the cobalt in the middle, the central metal, then you have an upper and lower ligand. The lower ligand is the DMB, the dimethylbenzimidazole, which is for the B12 active form is the same here, which could be for the pseudo B12 forms, could be different structure. It's known that some algae that they produce some different pseudo forms, and there are as well what we expect. There are many different other compounds around here from the evolution point of view here. That's so that the algae or bacteria, they developed as well other different forms of it, which about their bioavailability and let's say their vitamin B12 activity is nothing known about it. But we want to focus on the B12 active forms, which can be differentiated from their upper ligand here. And at the upper ligand, you see the adenosine cobalamine, the methylcobalamine, hydroxycobalamine, and the cyanocobalamine. Cyanocobalamine, which is the form that how it is supplemented, and as well, which is analyzed with a common general B12 analysis. But we have been looking at all of them. First question, okay, how do you produce the standards, the labeled standard, which is, you can imagine this is really tricky. We haven't done this by chemical synthesis, but by microbiology. So bacteria, we fed them with 15N, with ammonium, and they produced B12. Then let's say the tricky part of this really is to separate extract the labeled forms and to separate them chromatographically, which was really tedious. And then we obtain the forms given here. So we have them fully labeled here on the one hand, but we produce some partially labeled because the problem of if you just use the fully labeled ones, the problem is that, for example, the methylcobalamine degrades during the analysis to the hydroxycobalamine. And that's why then you couldn't differentiate between both here if you don't have a different label. And that's why we produce as well for the hydroxycobalamine a differently labeled one just to analyze this one specifically. Yeah. Right. Analysis, how does it work? So it's rather similar in the beginning to the other types of stable isotope dilution assay, with, let's say, one exception is here in the middle is the immunoaffinity column cleanup. So which you need due to, let's say, the small amounts that you need to analyze, and you need to increase the sensitivity of your method. And this is, for us, currently it's just possible here, only possible with the immunoaffinity cleanup, which is costly. So can expect that one immunity affinity column is about 10 euro, yeah, or 15 Australian dollars. So that's really expensive as well. Right. About how does the analysis look like? So just look at the right side of the slide here. You see it for the analysis for the pork fillet. On the upper side, you see the 4 different vitamers, which are clearly separated, well-shaped peaks with the hydroxycobalamin, adenosylcobalamin, cyano, and the methyl cobalamin. And then on the lower part of the chart, you see the labeled compounds as well. Nice peaks, and then you can rather easily then calculate here the amounts. Some results here of you see here for example the pork fillet, some meat samples of course are good sources for vitamin B12. So with pork about 20 nanogram per gram, to the lamb, which is an even better source with about 70 nanogram per gram. So if you consider which is what would you need? So the daily recommendation is about around 2.4 microgram per day. So that means you would need about for 100 gram of pork fillet, for example, here to cover your recommended daily intake, and for lamb fillet even less. Yeah, just about the validity of the method here, which is given with a pig liver BCR, which is a reference material where the certified reference value was about 1120 nanogram per gram, and our result give us rather the same value. So the assay is really valid. Right, then to conclude and give an outlook. So for B9, it's rather, as already mentioned, the bioavailabilities are very variable. So depending on the matrix, you see, for example, strawberries about 100. For the yeast, it's about 70 of that. And this as well is rather variable depending on the individual. So it's as well, we got at least plus minus 10% of different bioavailability. The general assumption of 50% is, I would tell it, not too bad. So it's a real rough number here, which we need to, let's say, further work out on it. For the short time methods, stable isotope methods are we think they are rather ideal. And what we still need here is really more intrinsic labelings here, really to have really correct models for the food, how the folates are incorporated in the food, not just by adding them or supplementing them. This does not reflect the reality here. So this is, well, our conclusion from the B9 results. For B6 and B12, well, we have been looking at food, and if we want to look at bioavailability and bioactivity, we need still the plasma analysis, and that's what we are currently developing, really, to go further. Right, that I'm right at the end of my talk. I would like to acknowledge here the group, of course, that is given here highlighted with Mangle Wang for the B12 analysis, Simone Jana for the B6 analysis, Lisa Obermeier for the B9 folate analysis. And I would like to acknowledge the traditional owner of the land here, because that they are really willing to share their knowledge and their wisdom with us here about their traditional foods. And as well, I would like to thank all of them and thank you for your attendance. And I would like to hand over here back again to Michael Netzel, the facilitator.
I'm coming. Just need to move. Thanks very much, Michael. It was a really great presentation. You know, I think about these analytical topics, which often are not really get the attention or recognition they really deserve. And yeah, now it's open for questions. And it's been settings. You just try to get the
probably short taskbar, yeah.
Yeah,
it should be here.
But we can't access here. We have
display setting, yeah. Change the setting. Yeah. Swap the view, possibly.
So we can't.
Here it was. You move a little bit the mouse and it comes to hopefully.
No. Oh, okay, yeah, great. Yeah. Now we got it. Sorry, guys. That's a bit tricky. Okay, there is anonymous attendee. Just curious if the bioavailability is so variable. How is the daily recommendation being set, Michael? That's a good question.
Yeah, tricky question. So it has been set by using, well, from bigger human studies here, and they are quite long ago have been done by not by short-term studies, by long-term studies, using a bigger, let's say, set of individuals. So that are not those 5 that we have been using, but they have been using up to 50, and so they consider that the individual variability is covered by that. Of course, you can still argue that the different food variability in the bioavailability is not covered with this, and this still is, let's say, we have to confess, yeah, and this is already, yeah, is being confessed that this is not different food variability is not really exactly considered. So we need to know, or we need to remember that this is really a rough number due to this variability, which we as well found out, and which is due to these constraints that there are, is because we don't have the, let's say, the variability, the human testings for all these different types of foods here, where we need to know the food, the folate content or the vitamin content, and then the plasma response for that. You can imagine, so those human studies are tedious, and those years or decades ago that there haven't been those folate or vitamin analysis available. So that's why we're just now in the process here, going with the better methods, with the new models and so on, to get a better knowledge. So yeah, keep in mind, so these numbers that are here being presented, or with the recommended daily intakes, they are rather rough. Yeah. So in the end, if you want to, let's say, want to check your status, yeah, so you can't really rely on the, let's say, on your daily intake, or if you have a collection, or you put a collection of what you have been eating and taking some databases and so on, and calculating your intake. This is really rough. For checking your status, you need to do really plasma analysis for this. And if you think, or you think that you're low in status, just have that checked with plasma value. I hope this answers your question.
No, thanks. Yeah, thank you very much. The answer. So just I have also a question, Michael, and you mentioned this is the bioavailability, in principle, 2 questions. So the one is the bioavailability, which I think you had one slide when you said mentioned is that it's still, but I don't think it's valid anymore. That food folate is regarded as a bioavailability of 50% compared to folic acid, which looks like is much higher, depending on the food and maybe also the method. So that I think in the future maybe they will have to not to rewrite books, but I think there will be some changes, because it looks like there's definitely much higher bioavailability. And the other one, how maybe to demonstrate how important the work is what Michael is doing, and also to have a suitable or a good, reliable analytical method. Like when you look in the nutrition tables and worldwide, in Europe, I just know from Netherlands, the German one, even the US one, and especially here in Australia, the Australian food composition database. And when you look, there is still, like for strawberries, a very low content, and it's very similar, like in the other countries, between, let's say, 39 or 37 to 45. So we just did a little bit in one of these, in the Natural Nutritious project, with you together. Would say a small range of mainly Queensland grown strawberries. You could find much higher content of folate than in the labels, like sometimes 3, 4, 5 times higher, and you presented this like 160. So it looks like, because when we talk to EFSA, so that's the regulatory body here in Australia, who is putting these recommended dietary intakes, and also the food concentrations and creating or updating the nutrition tables. They told me that they still, I think at that time, used the microbiological method, which Michael knows is much better. It's not so specific, and you can overestimate or underestimate, or calculate, a value. So that's, I think there is a really lot of work to do, and very important, because, for example, what you said, a punnet of strawberry of this high folate strawberry can easily cover your recommended dietary intake, or even let's say a standard commercial one, which around maybe 100. It's almost, if you eat a punnet per day, which is easy, you can easily embed this to a diverse diet. That's not something that Michael said, it's extreme examples, like you have to eat 4 kilograms of bread, or you have to drink 2.5 or 1.7 liters of beer, something. A punnet of strawberries, 250 grams, a serve in the morning, serve in the afternoon. That's really doable. So that could be not a game changer, but, make also this, a more healthy or diverse diet much more attractive. If you have, can you link this, for example, to maybe the real, inverted commas, real folate content. So Mr. maybe you can
right there, two questions, aren't there? First I have see a further one in QA, but I try to answer the ones from Michael. So but a 50%, yes, indeed. So we have, from our results, we found what I have been presenting is that for the strawberry it's almost 100. For the yeast it's about the 70%. We found as well for some cheeses, and what was rather surprising to us is with some camembert cheese, on the one hand we got around, I think when I remember it well, it's about 60% on one type or one brand, and the other brand had as low as below 30 as well. So at the current stage, I don't feel that confident that I can really change the 50% currently, or recommend to change the 50%. But it would take lots of more analysis and human studies, and Michael, that's tedious, and we would need for this the funding of that, which I see currently funding for this human study, which I don't see currently really being favored currently, because I think the governments, they think they're doing it easy. They just do mandatory fortification with folic acid, and they say that's it. Yeah. But I think Michael and myself, we are not in favor of. We want just do it naturally here, really what is in the food. So that's why for this really to have these numbers change, we would need more funding and more studies, human studies for that. The further question about let's say how these values or new results of the high amounts, how they can be really included in the databases. And what you correctly said, Michael, is about the reference method. And often those databases they just accept reference method, in which for food folates is still the microbiological assay, which we don't use here. But what I can tell you here, just in Germany, there is now an let's say a collab study being let's say in preparation here, where I am involved as well, where the isotope label, a stable isotope label assay, which will be tested as well, and to implement it as a reference method. And that could be rather a game changer for that, because this would be the first one, really the first collab study having a first reference method for it. I think that was that.
Yeah, no, thanks. No, I think that would be really good. And I think, and it's a question, Bernardine, just a second. And that's what EFSANS, and they were really very open for new methods. And I think they're aware that the microbiological method has these really significant limitations. But what you said, it's always, if it comes to the point, the funding and, how big should the study be and all the, especially for this one, you need a special sampling design. And so it's not that easy, but I'm sure we will have a change in the near future about, even the nutrition table, not just for strawberries, maybe for so many other foods. Okay, now is a question from Bernardine. Thanks so much for your presentation. Is it possible that some people are not able to produce the methylated folate from folate? So a physiological question, Michael.
Well, I'm not a physiologist, but yeah, it's right. So there are different aspects. As on the one hand, there's a genetic, let's say, genetic issue for the let's say the methylation process in the physiology and there are some genetic disposure of that, or let's say some genetic status where the people are really compromised with a folate physiology, which is an issue, of course. So I think it's relevant for about 5 to 10% of the population, in which when we do our human studies, we screen for that. And I would say this is important, particularly I would say just for women in childbearing age, which are plan to be pregnant and get children, to get themselves tested, because for those compromised people, they need to have a much higher intake of folate. Yeah, really supplementation, really, because then it's well known that these genetic forms, that they are highly more probably to get the neural tube defects. There's another aspect is of the, let's say, from folate, from let's say folic acid, from the supplemented form, and it's known that there are some, let's say, you find in consumers, you find the non-methylated form of folic acid, which is in discussion, being, let's say, carcinogenic and so on, which as well is an issue, let's say, in the discussion about the folate fortification. And probably, well, one solution would be if fortification would be done with the 5-methyl form, which currently is not being done. So there are recommendations for doing that, but the industry is not really keen on that, because the 5-methyl form is not that stable, and it's more expensive and so on, and that will change a lot probably in technology, and that's why. But I think Michael and myself and all of other folate researchers, they would really be happy or be to change from folic acid to 5-methyl tetrahydrofolate in supplementation. Like some comment from your side, Michael?
No, I think you answered this, with the genetic disposition and for 5-methyl, and also you know, it's when we did our study, it was all really difficult to gather you know, a non-folic acid supplement, you know, like the 5-methyl, and I think that's, you know, that's a bit of a problem, that you're still like, you get folic acid and fortified, and often, you know, in these supplements, it's only, and it's very highly dosed. So if you look at the dosage, it's really, really high. So, and it's still this mentality, you know, like these supplements, often the people think the more the better, which is not always. It's not just for folate, it's also for vitamin C and others, you know, and they take them instead of 1, 2, or 3, you think, wow, it's great, you know, it's better. And that's also something, that's why I think the idea of having a natural source like fruit or veggie, which can deliver most of your nutrients, and would be a very good alternative. And I'm not saying that you should, like, if you now pregnant or getting, you know, or you get this with your doctors, you know, follow for sure, you know, the advice, but it's something, you know, like part, I think, could be part or should be part of a healthy nutrition. Like, you know, like if you look for high fiber, you know, food and these things, you know, and I think that would be, it's something, you know, for the future, I think, you know, we should really, you know, keep in mind. Unfortunately, because of the time, if they have to finish the Q&A session, but stay on, we have 2 more slides. Please, if you have any questions, what I said before, or interested in something, just send Michael an email. If you don't have his email, just send me an email, and I will forward it to Michael, and I think he's more than happy to answer your questions. So I just have to, and I apologize for some technical, because we have a big screen in my small laptop screen here at Elkhorn, and it was a, hang on. We had a few, perfect. Now it's going. Sorry. Yeah, it's because it's tricky with the mouse between the big screen and the laptop. So, I want to promote also next seminar on Tuesday, 28th of November. It's from Michel Bayer. He's a PhD. He's almost finished his PhD. Also at the Center for Nutrition and Food Sciences, and he's working or still, you know, finalizing on kakadu plum fruit bioactivity against spoilage, microorganisms, and oxidative reactions in refrigerated raw beef patties, or like in burger patties. And he will present his really exciting results on the 28th. So please, if you have time, zoom in again. I think it's really worth. And the last slide. So if you, again, you know, really thank you very much for attending. And if you enjoyed today's seminar, just sign up to our seminar invitation list at quafi.uq.edu.au/science-seminars. And I think that's all from us. And hopefully enjoyed the seminar, and see you then next time. Thanks very much. See you then. Bye.